and then we will move on to saying hello to everyone. And welcome to our February meeting. Hopefully everyone's enjoyed the wonderful weather today anyway, and been out and enjoying things that are going on in this time leading up to, of course, springtime just around the corner. So as we always do, we'll talk about some bird sightings, have a really easy identification challenge because there's a lot of news coming from the club this month. We'll meet up with our committees to discuss anything going on. We'll talk about the field trip coming up in a few days, open any discussions of things that we have not talked about. Pete Peterman has some exciting news about things coming up in the future. And I know Bilbo has some things to talk about with the Christmas bird count. So we will address those. And then our monthly presentation related to conservation of waterfowl. So please feel free to start to share some of your bird sightings. Pete, I do have the image that you sent me after this, 2 more slides from this, because Diane also sent a wonderful picture. But if others want to share what they have been seeing or hearing or what's been going on as they're out and about, please do.
Been up in Deltaville the last few days working on our new JRA boat. It's a dead rise up there, and we were out on the bay today, and it was loaded with long-tailed duck out on the bay today. So hopefully we get some stuff this weekend.
Anyone else?
Sean, is that a recent picture by Katrina?
Yeah, just a couple weeks ago. Because my yards are open, so I collected some brush so that Cooper still keeps coming by trying to eat all my birds, which I don't mind because they're mostly starlings and blackbirds and cowbirds.
So it looks like a juvenile.
Yes, we had an adult, but this is who's hanging around right now the most.
This is Angie. I was walking yesterday at Riverview Farm Park and saw 2 meadowlarks, eastern meadowlarks, which I've never seen over there before.
All right, good bird. They're a pretty bird.
This is Phyllis, and I still have the 2 hummingbirds, amazingly enough. The ruby throat female usually comes to the back feeder, and the we're calling a rufous male usually comes to the front feeder. I didn't know if they'd make it through last week. We weren't here, but one of my neighbors warmed up the liquid for them, and they are still here. So we're happy for that.
This is Twyla Bloom. I was also at Riverview Farm Park recently, and I saw some beautiful light yellow birds, small, and I looked up and I think they might be American finch. Does anyone know? Is that common around here?
Yeah, so they could be American goldfinch, not too terribly bright, but kind of a yellowish, dull looking color right now because of still in that winter plumage. That seemed to match what you were looking at, whether one is bright yellow and black capped as maybe the images, but kind of still yellowishy.
Yes, they're, I would call a pale yellow with gray, maybe on the wings. I also saw these same birds at Mobjack Bay Coffee Roasters in one of the trees there. There were many of them there, and the sunlight would catch them and you could see the yellow. They're just small birds.
This is
Yeah.
Barb. I'm very happy that I have a ruby crowned kinglet that's hanging around. I've been seeing it for several weeks now. Usually I see them once or twice and then they're gone.
We're down at the Sandbridge and we saw a raft of probably 200 or 300 red-breasted mergansers right off the shore.
And this is Pete, and we are again hosting a whole bunch of vultures. They all form in the park, which is across the water from us, and then come over to our property and sit on the house and in the yard. And the majority of them are blacks, but there are a third of them are turkeys. And the number of turkeys and blacks change each day, but they continue, and this has been going on now for the past 2 weeks. They seem to like the environment because the sun comes up and they can sit in the trees and spread their wings and get them dry. So yes, vultures are here again, just like last year.
That's really cool. A little creepy. That should keep salespeople away, but that's a cool thing to have going on. So let's move. Diane and Jane were out last weekend, according to the email. Was able to see a beautiful painted bunting. I don't know if Diane's here this evening.
Yeah, I'm right here. A lady who had been coming to some of our walks last year lives over in Norfolk, and she texted Jane about this bird. And if anybody is interested, it's still hanging around her feeder. You can give Jane a call and she can give you the information. The lady doesn't want it out publicly, but she welcomes members of the bird club.
Awesome. It's such a very nice picture. I know it's a little bit grainy because I had to blow it up and it came through the email, but it's really composed well. It's a beautiful picture. So thanks for sharing, Diane.
Probably it's probably an ASY, a 3-year-old male. It's not an old male painted bunting because the red eye ring is broken. I used to band painted bunting, so this is probably a recently molted into adult male plumage bird, probably a 3 or 4-year-old.
Oh, that's really awesome.
Thanks for that information because we were wondering about that.
Yeah. You can't, with the green birds that show up, you can't necessarily tell from a photo sometimes, but you can't necessarily tell a green painted bunting as a juvenile male or a adult female sometimes because they look very similar.
All right. That's good. Thanks, James. Love learning things from you. Pete, your picture, yep, your picture is next. And then after that, Pete, I have part of your checklist if you wanted to share about that as well.
Yeah, so this is a leucistic common grackle. The week before, I had a leucistic red-winged blackbird male. And so this guy showed up, and as you can see, he's got the golden eye, and other than his head, he's got the coloration of a normal common grackle, but he's kind of a weird guy. The other birds, they weren't particularly happy with him around there, but he seemed to be able to get food and everything. So this is him. And would you like me to talk about this? Okay, great. So this past weekend was the Virginia Society of Ornithology's annual trip to the Outer Banks. Now, that trip hasn't occurred. The last time was in 2019. So this is the first time since COVID that we had the trip. And if you've never done it before, I would highly recommend it. There are fabulous birders in Virginia, and VSO has them there. And it's just wonderful for somebody like me who's not a fabulous birder to have somebody who can help me in identification of certain species. But we had over 130 species that could be seen. Some of the ones I did get a lifer in a woodcock. I had never seen them, and we were out at night for the short-eared owls at Alligator River, and there were a couple woodcocks flying up in the air and then slowly coming back down and doing their buzzing call. So that was very fun. There was an ash throat flycatcher, and the last time I did this tour, we got an ash-throated flycatcher too. So it seems like about this time of year, they're not supposed to be here. They're supposed to be out west, but there's plenty of food for him in Alligator River, so he's there. Alligator River also had rough-legged hawks, which I got to see. And at Body Lighthouse, Bob Ake found a Vesper sparrow, which is something also that is not a normal thing to see. So over 130 species, lots of tundra swans, all the ducks that you can have there. Friday at Madam, and then in Saturday and Sunday, it was Arctic. Winds 30 knots, cold as can be, very difficult, but the birds were there, and so if you just dress accordingly and you can see many birds. So it's always the first weekend in February, so think about it. It's a fabulous time. This was completely COVID related. We had no indoor meetings. Everything was outdoors. We did a Zoom meeting before to talk about it. And so it was a great experience. Do you want me to cover what's coming up at this time, Sean?
I have a slide just for that with that new business.
Okay, thank
Yeah, no
you.
problem. So let's do our quick identification challenge. All super easy things. Now I always say it's super easy, but this time I really mean it. It's going to be pretty easy. It's things that I've been seeing around. I know we're not stumped already.
Canada geese.
Yeah, so just some Canada geese that are out back in I'm liking them because they're a little bit smaller, not like the resident guys. And just I've really been enjoying them and watching their behavior. Here's another group of birds that I've seen a couple of times when I've gone out to Buck Row to go to the playground. Well, not me, Henry's going to the playground, but I'm birding while he's doing that.
Looks like Forrester's terns.
Yeah, it might be hard with the picture, but the Bonaparte skulls.
What?
And then one more, our last one for the day.
American black duck.
Yeah, so I think it's an American black duck and mallard hybrid. Is this
Yeah, it looks good for hybrid.
So given the theme for tonight's talk, I threw that one in there for everyone. So I don't know if Wen is here, and I forgot to ask if there was any new members tonight. As people were coming in, I didn't notice anyone, but is there anyone new who hasn't joined us in the past?
Not that I know of, but John Adair usually keeps me posted, so.
Yeah, and I know John's away. Tom, do you want to say anything about the newsletter?
Yes, this is Tom Charlock. We'll have a newsletter coming out at the beginning of March. So please send inputs to me by February the 25th at the latest. If you have notices to the club, count results, or especially I'd like to see our photo contest winners from Michael and any contributing authors. If you have a secret spot in the area where you go all seasons and find lots of birds and you haven't told us about it, just send in a paragraph or a couple of sentences about it, and we can spread the word out. So thank you.
All right, thank you. I don't see Mike. I know the Maxis have been doing stuff with the webpage. Ellis or Wendy, do you want to say anything about that?
Really have much to say today. It's
That's okay.
very helpful when people have something to post that they go ahead and send me an email directly. Sometimes I pick it up in other people's conversations, but it's sometimes hard to get the information.
Yeah, so I think Ellis makes a really good point, because I know we all chit chat about things and it gets filtered to Ellis, and he's mentioned this a few times now. So I know I'll certainly be a bit more mindful, making sure I send him things directly myself. Field trip this weekend, James. I think I have the details correct. I did find it in my spam folder.
Yeah. Still gonna keep plugging along. They Back Bay pulled the plug on tram trips on Monday, so we'll get another shot at Grandview. Weather looks good this time, so just like Grandview was going to be, we'll meet at the entrance gate at 7:30 this Saturday. You park there on the right hand side, and then we'll go into the beach, and feel free to walk as much or as little as the beach as you want. It's a long way to get all the way up and back, but dress for the conditions. I think it's going to be pretty nice, though, not like a normal Grandview trip where we're freezing, but it can definitely be a little chillier and windy out there. I'm hopeful that we'll see good numbers of waterfowl. The bay is pretty loaded right now. When I was on the boat, and Grandview was pretty good the other day. There's been some Harlequin ducks, male and female, hanging out by the lighthouse ruins, so maybe they're still around. But yeah, my contact information is good, and we'll see you there. So no Back Bay tram. Keep trying at that. I also heard from Craney Island they pushed back their field trip decisions until March. So I'm still trying to get that one for April, and we'll see. But I'll keep trying to get some field trips going again. I'm also working on potentially next winter getting our driving tour of the Chesapeake Bay Bridge Tunnel back. I have an inside track on maybe making that happen, which is exciting. So we shall see.
And thank you, James, and thank you for working really hard this, I was going to say academic year, because that's how I live as a university professor, but this year.
Why, as an
dealing
environmental
with-
educator.
dealing with COVID and trying to plan things. And also, you know, it seems like Mother Nature has been hitting us on the weekends with some nasty weather instead of giving us school days off, that weather during the week. So we appreciate it. Thank you. Pete, I know you wanted to make an announcement about the VSO meeting. And then Bill, after that, your Christmas bird count. And I'm just trying to keep us on track so our speaker can start relatively close to 7:30, so we're not taking up too much of his time.
Okay, so the announcement is the VSO annual meeting. In 2019, there were some of us who were working with Williamsburg Bird Club to put on the annual meeting for the Virginia Society of Ornithology. And of course, that got canceled because of COVID. So 2020 was canceled, 2021 was canceled. So what they're going to have, and Bob Ake is really the spearhead of this, with the Cape Henry Audubon Society and the Friends of the Dismal Swamp, they're planning a modified VSO annual meeting and a Dismal Swamp Birding Festival the weekend of April 29th to May 1st. So there's not going to be anything indoors, so you don't have to worry about that. There will be a meet and greet, hopefully, if the weather is good, at the hotel. And the hotel is the Hilton Garden Inn, Suffolk Riverfront. And all this information is going to be on the VSO website, which is virginiabirds.org. So I'm to announce this to you that after 3 years, they're going to have an annual meeting, and there's going to be bird walks in the Dismal Swamp, so you know the birding will be good. It'll be springtime, so there'll be lots of warblers. So if you have that time in your calendar, it would be wonderful to see you there.
Great, thank you, Pete. Bill, I have your slides, and just let me know when you need me to advance them for you.
Okay, just a quick summary of the count that we conducted this past December. Total species 115, total birds over 20,100. We had 40 field observers, which was a few more than last year. Only 4 feeder watchers, which was a little lower than I hoped. And 16 field parties. It wasn't a great day. It was at least warm, but light winds, but morning fog and light rain. So the fog, I think, really inhibited visibility for the ducks, which they were pretty few in number, the sea ducks, particularly the offshore birds, probably because people just couldn't see them. We only saw about 2,200 of those ducks versus normally 3,500. We saw no northern gannets and no fox sparrows, so that was kind of unusual. Next slide, please. So we had some interesting observations this year. The first ever black-throated blue warbler by Mark Nichols. There's a picture of that bird. It's in his yard. It's gone by now, but it was there for a few weeks. We did have one black-crowned night heron in Chesapeake, and you can see that photo is there by Laura May. In Fort Monroe, they saw 4 brown creepers on the same tree. And some days you can't see even one, and they saw 4 at one time. Some tree swallows, which are uncommon, salt marsh sparrow, the pipits at Grandview. And of course, Phyllis had her ruby-throated hummingbird, which was the only one we had. And we had some all-time high numbers and observed for the bald eagle, which probably isn't a surprise, which is great. Clapper rail, yellow-bellied sapsucker, downies, white-breasted and brown-headed nuthatches, eastern bluebird, and black and white warbler all were all-time high numbers for those birds. Next slide. So just a quick overview of the level of effort that was undertaken. No owlers this year, and obviously we had fewer hours at feeders because we had fewer folks doing that. Next slide. And again, I always like to do this shout out for everyone that participated. Many of you are many times doing this for me, and I really appreciate your efforts. Folks with an asterisk are new this year, including Laura May, who's here today or present virtually. And next slide. The number of individuals, here's our trend. I start this in '93 because that's when we really started consistently surveying all the sectors we have. The trend is still decreasing, not quite as much as last year. You can see that little uptick at the end, but generally not a great trend for us here in this circle. And the next slide. So the number of species, a slight increase, and it is very slight trend going up. So I don't know if it's really very significant from a scientific basis, but if there is a trend, that's what it is, and it's not very marked. And next slide. Just a reminder to all to do the Great Backyard Bird Count next weekend. So I think most of you are familiar with it. So please participate. If you don't know how, shoot me an email or just go to birdcount.org, and they've got everything there. There's even a webinar this week coming all about the bird count and how to do it. So that's all the slides I have. I would like to announce, get an early marker down for our spring bird count, which I was going to have on the 30th, which we normally do the last Saturday in April, but not to compete with the VSO meeting. We'll hold that on the 23rd, April 23rd for the spring count. And that's all I have, Sean. Thank you.
Okay, great. Thanks, Bill. And just as a little side, Bill, and since everyone's listening, has shared some of those spring count data going back to, I think, 1995. And I had a student working on that, then COVID hit, but I have another student who's entered in a number of those years, and her project this semester is going to try to pull out some of those patterns and look at population trends, and also do some GIS work to look at land cover change or population growth, and see if we can talk and tell a story about all the data that you've all been collecting. And another student's working with the data that we collect each month or twice a month. So I'm just letting you all know that you're helping to provide students with some research experience, and I'm trying to get them to go out and join us. And I can't take a van because of COVID, there's some other things, but I'm trying to get everyone to come out so we can kind of interact and get more students in the field. So thank everyone for doing that. So this evening, I would like to introduce Phil. I don't think I spelled your name like Loretsky, assistant professor. Sorry, I'm sorry.
There you go.
assistant professor of biology at the University of Texas, El Paso, where I did my doctoral work. He's also the curator of birds for the university's biological collections, holds a BS in genetics from UC Davis and a PhD in environmental science from Wright State University in Ohio. He's held positions as visiting researcher with the Smithsonian and as a biological science technician for the USGS. His work has focused on understanding the nature of waterfowl population genetics and evolution. Tonight, he'll discuss Mallard begets Mallard. Population genomics help shed light into the outcomes of secondary contact and its consequences for conservation of several duck species. Phil, I hope I've made you a co-host and I'm not doing anything stupid that way. I'm stopping sharing, so you should have complete control.
All right, perfect.
Yep, and thank you again for taking time out of your schedule to meet and share.
Yeah, absolutely. All right, can everybody see that? Let me minimize this little.
Yes.
All right. Trying to move some of this stuff around so I don't see it. Nope, doesn't want to do it. All right, we'll go with that. Yeah, Sean, thank you for having me on the internet with everybody. I'm going to talk to you a bit about what my lab does in my research program, and happy to answer questions. In general, my lab does a bit of everything. We do everything from, I say, gene to field. And so we try to get kind of this like all around understanding of the populations that we study. I generally call ourselves a wildlife genetics lab with a focus on waterfowl, but in essence, we work for anybody that's got DNA and money. And so we can answer those kinds of questions that they have, everything from, snails to whales to ducks and birds and various other things. Again, the students that go through my lab, I try to get them all the kinds of experiences so they can actually talk across fields from ecology, biology, genetics. On top of that, I make sure to kind of impart our knowledge at the international scales, whether we're in South America or New Zealand or Canada or Mexico or elsewhere. And then on top of it, I'm really keen on ensuring that the biological collections at our museum here and elsewhere, we share materials with lots of different museums like the Smithsonian, and make sure that that's going on. And of course, community engagement is a big thing that my students and I do. And so I'm happy to always chat with those that are willing to listen. So in my lab, so we are a genetics lab, we do everything from single gene to partial genome to full genome. And that's really just depending on the question that we're trying to answer. We might look at a single gene, we might look at parts of the genome, or we might look at the whole genome, depending on, again, what those questions are. But the primary method that we use is called dd-RAD-seq. It's essentially taking the genome and cutting it up in a certain way that we can consistently gain markers across the genome for species for anything really, for any non-model system. So I've used this everything from lizards and snakes to birds and mammals, and it works. It continuously works, and we get a nice snapshot of what the genome looks like. So all the data that you're going to be seeing here is basically coming from this method, and so you're looking, the analyses that we do now are, instead of like one or two genes, we're looking at thousands of them all at once for, as you're going to see, anything from hundreds of samples to now a couple thousand samples that I'll be presenting on. And what do we do in my lab? It's really population structure. My lab is both evolutionary focused and more applied. And so but essentially, whether it's evolution or conservation management of populations, it's really asking the question of what are we working with? And that's why people come to me, why agencies come to me, is they essentially want to know, what is this thing? Is it a population? Is it a new species? Is it a subspecies? What is this? And so that's what we try to do, is essentially understand those populations, what their true evolutionary history is, and that essentially guides better conservation and management, because if you don't really know what you're working with, if you think it's a species, but really it's some sort of population or vice versa, your strategies might not actually be effective. And one of the key aspects that we study in my lab is hybridization. And so given that all of you folks know birds hybridize readily, but in fact, really, while we always thought that most lineages would be bifurcating where one species turns into two, as we continue to gain more and more genomic data, we find that many lineages have intermixed genomes, including humans. If you thought the Homo sapien genome is its own thing, it's actually pieces of many old human lineages, from Neanderthal to Denisovians, depending on where you're from, you'll have a bit of them as well. So there's been hybridization amongst many lineages, and that's an important thing. But why in birds? Well, birds hybridize quite readily, even though they're one of the oldest lineages. They retain the capacity, many of the species complexes retain the capacity to hybridize. We think that the reason that there is such a high rate compared to mammals or plants or inverts is that they have strong dispersal ability. They're able to move about the world. And for whatever reason, their chromosomes, the way that the genes are mapped across the genome, have been quite static. And as long as the chromosomes are the same, it is very highly likely that you're still able to make viable offspring. So they're able to move about. They have these chromosomal the genome is very stable, and they have relatively low levels. And those two things is why potentially they have relatively low levels of reinforcement. And reinforcement means two species put together, they still make viable offspring. If it was truly reinforced, they would not either identify each other as potential mates, or if they did, it would be more of a horse, donkey, mule situation where the offspring are infertile or inviable. The tree of bird life is here, and the plots that you can see here are essentially the rates of hybridization. How many species or how many taxa within the order can hybridize? Here is in Seriformes, and 60% of waterfowl species hybridize in nature, and if you put them into a cage, you can increase that up to 77%. And that is across ducks, geese, and swans. All of them will be able to hybridize with it, not obviously geese and ducks, but within the swans, within the ducks, and within the geese, you can have quite a bit of hybridization going on. And this is where I usually stop, and I want to make sure that everybody's on the same page of what hybridization is and what gene flow is, because I'll be talking about these two aspects. And hybridization is this, where you have parent one, a mallard, parent two, a black duck, making a hybrid. And if that hybrid does not breed back into parent one or two, that is essentially lost breeding potential. And hybridization occurred because you have this F1. But if that F1 is inviable, it doesn't breed, then there's no real movement of genes, and essentially lost breeding potential of parent one and parent two. Gene flow is hybridization, but then that entity, right? You can think of the F1 hybrids as a vessel that holds the genes of both species, and that vessel needs to breed with parent one and or parent two to actually move the genes between species. So gene flow is hybridization. Hybridization occurs, and then you have back crossing. So that F1 actually breeds with the parents, and that actually moves genes between them. One of the focuses of my lab recently has moved from wild interactions to domestic wild interactions, and what is coined anthropogenic hybridization, which is the human-induced secondary contact, or human-induced hybridization, is becoming more common. And so this is the direct or indirect movement of pen-raised individuals, increasing the presence of feral populations, and thus the gene flow with their wild counterparts. So we can think of rainbow trout, or really salmonids, any of the salmon, any of the fisheries that we pump fish that are being stocked, that is a form of anthropogenic hybridization. Feral goats, wild goats, pen-raised bob white quail, and wild ones, pheasants, pigs, so forth and so on. All of that leads to higher rates. All right, so I want to introduce you to, so that's kind of the background, and I want to introduce you to the main group of organisms that I use to study and understand hybridization and what's occurring and what that means to the species and to conservation. And that is called the Mallard Complex. If you don't know this, there's about 14 mallard-like ducks all over the world. Outside of the mallard, which has a whole Arctic distribution, and in fact, pretty much worldwide outside of Antarctica, the rest of them are endemic to regions or islands or locations, and they're really only found there. The other important aspect is the mallard is the only one that is dichromatic, meaning males and females have a different plumage. The rest of them are considered monochromatic, where males and females look very much the same. Amongst this group is that I'll be focusing today is called the New World Mallard Complex, which is, of course, those birds that are found in North America. And that would include the Mexican duck, which is found in the Chihuahuan Sonoran Desert, where I am right now, the American black duck, where you guys are. The mallard, well, the mallard's found everywhere. And then the two populations, or though I think they're now subspecies of the mottled ducks, which are West Gulf Coast that are found, that range across Texas and Louisiana coastlines, and then the Florida mottled duck, which is specific to Florida. So the history of this system, if you don't know, all of the birds outside of the mallard are called monochromatic ducks, meaning that they are very much, the males and females again display the same plumage type. But we know, and I saw the discussion of those black ducks, that one of them potentially is a hybrid. And that is because there are characteristics within the monochromatic ducks that everybody in the dogma that has been perpetuated for a long time is that mallard-like characters indicate hybridization. However, there could be an alternative hypothesis, which is that the reason that they show it is not due to hybridization, but due to the fact that the mallard might be their ancestor, and thus it's really just the vestigial character or the remnants of their dichromatic ancestor. Of course, everybody for the last however many years has gone with hypothesis one, that is hybridization. And so hybridization must be a problem because we see lots of these ducks showing mallard-like characteristics. And until recently, genetics has not been very conclusive. And that is because genetically these things look the same. And that was, of course, used as additional reasoning that it must be hybridization. American black ducks, mallards, and Mexican ducks, I couldn't even tell them apart in 2014 with 20 loci. Of course, Florida mottled ducks, the mottled ducks actually are quite distinct and you can find them. But if you can't tell the parents apart, well, either the parents are dead, they're genetically extinct and everybody's a mallard, or we just don't have enough resolution. And so as new methodologies, genetic methodologies, came to be, I, of course, wanted to apply them to the system to understand whether increasing the number of loci would actually give us any sort of resolution. And in 2019, I published a paper on this group, right? In 2014, I did it. In 2019, add more loci, add more samples, and voila, there they are, the parents. And in fact, by increasing the number of genetic markers 200-fold, you can actually identify individual species. Now, if you've never looked at these sorts of maps, these are called assignment probability maps. If you've ever done 23andMe or Ancestry.com, it's kind of like painting your chromosomes based on the ancestry calls. And this is the same thing. If an individual is assigned to one color, that's one population. If it's assigned to multiple colors, then it's potentially a hybrid because it has ancestry of multiple populations. All right, so with that, I'm going to be showing you more of these types of colorful ancestry maps. And so we say, okay, so we can identify populations. Let's go and go into each one of these groups and see if we can't find all these hybrids that everybody thinks that are out there. So where are the hybrids? So first, for Mexican ducks, we expect lots of hybrids throughout much of their range, as even in 1970s, Hubbard suggested that they were on the edge of genetic extinction at that point, and that really resulted in the de-elevation of Mexican ducks from Anis diazi, from species to subspecies to Anis platyrhynchus diazi, which then wiped the hands of what Fish and Wildlife had to do at that point for this group, because it's like, well, it's a mallard. And so this is what we've got, right? So again, lots of coloration here. I want to note on the left, so over here we have domestic lineages. The black is a khaki Campbell, and you'll see this throughout the talk because I use this to understand potentially hybridization with domestic lineages. And the red are game farm mallards. These are lineages of domestic mallard that are bred for shooting purposes. Excuse me. So what can we see here? So we have domestic mallard. The green is wild mallard. And then the rest of these are Mexican ducks collected at various different locations. And the north is US versus Chihuahua. The West Coast is Sonora and Sinaloa, and interior is essentially Durango, all the way south to Puebla. So the first thing that we show is that there are low rates of hybridization between Mexican ducks. In fact, it's only about 5%, very similar to pretty much any species bird species complex. This is in fact 0% in West Coast and interior. US birds have the highest ancestral genetics of all the other populations, and it's actually not due to hybridization. We do lots of different stuff. We do different analyses to really understand whether all that green in there, that bit of green, is actually hybrids or ancestry, and in fact, it's actually due to ancestry. And so what this suggests is that potentially what this suggests, and more of our research, is that the mallard essentially came, you can imagine the glaciation pushes mallards down. There's a pocket of glacial refugia in the southwest. As the glaciers retreat, mallards retreat, but then a subset stay in the north and then essentially lose their green head, and then through movement southward populate the rest of the Chihuahuan Desert. And that's what our findings suggest. What about mottled ducks? So mottled ducks, again, endemic to the West Gulf Coast and Florida regions. Again, expect lots of hybrids. However, when we did that, we actually didn't find that. We found that both the Florida and West Gulf Coast mottled ducks are as different from each other as they are from the mallard, and that hybridization is once again 5 to 9% in general. And so, and the nice thing is that there's another study later on, many more samples, and they too, by Ford et al., again, finding the same exact results. So that's nice. Again, only about 5%. All right, now moving to American black ducks. So this is a more complicated situation, and you're going to see several different analyses. So there's a few things here. The first thing is that American black ducks, genetically pure American black ducks, exist. The problem is how U.S. Fish and Wildlife calls their American black ducks hybrids and mallards. So only 80% of those black ducks that were called phenotypically based on the wing plumage were actually black ducks. And the exact same proportion of mallards were correctly called as well. That means 20% of black ducks and 20% of mallards being called in general by Fish and Wildlife are actual hybrids, but are being called pure, parental. The worst of them is in the Atlantic flyway, where there's actually the most amount of hybridization, but only 70% of mallards in the Atlantic flyway were correctly called. Of the hybrids that exist, 40% of them were incorrectly called. So it's essentially a flip of the coin. Almost 60% of hybrids were correctly called. 29% of those should have been black ducks, and 12% were actually mallards. So what this tells me is that the current sweep of the traits being used by all of us essentially are a flip of the coin and are not actually indicative of a hybrid. They are not true phenotypes that say, yes, this is a hybrid. And I want to also point out that in model ducks, where that white wing bar is used as a hybrid, when we did this study, in fact, 10% of pure model ducks show that trait. And so that actually suggests that it's just in the population. And of course, if you didn't know the genetics of them, you would be like, oh, well, 10% of those are hybrids then. But in fact, it's actually a formative trait. Same thing with black ducks, and same thing with Mexican ducks. Many of these mallard-like traits come out in the formative plumage. All right, so what about hybridization? So, okay, so phenotypic characters, not so great. What about hybridization now that we have all this? We have lots of genetics on them. So 25% of the samples that I had were hybrids. That's quite high. It's actually the highest of that group. And the proportion of samples that were identified as hybrids increases west to east, which makes sense. But there's also this interesting pattern. So if you look at the mallards, you see that there's these two populations, this gray and a black population. The gray, so all mallards that I sampled west of the Mississippi, called west, were this gray population. But as you continue to go eastward, more and more of these birds were identified as not only carrying what we coin Old World A mitochondrial haplotype, meaning a Eurasian derivative haplotype, but also a non-Western mallard genetic signature. So what is happening in Eastern North America, right? So 74% of them carried a Eurasian lineage and this non-Western mallard genetics. So if you don't know your history of Eastern North America, this is what happened. So in the East Coast in the 1920s, people were like, hey, I would like to shoot greenheads, but greenheads were vagrants and very few and far between in the Atlantic flyway. And so folks were like, okay, let's just captive breed some birds and throw them out. So they started with black ducks, that didn't work. And they started with wild North American mallards, that didn't work. And somebody had the idea of going to Europe and picking up game farm mallards that people have been creating for the last 100 years, again, in the 1920s. So from 1920 to 1960, they brought them, they propagated them, and state, federal, and private individuals released about 500,000 of those per year. The other part of this story is that Western mallards started to creep into the Mississippi and Atlantic flyway, so crept further and further east as the boreal forests of Ontario and Quebec started to be cut down and start to look more like a prairie where they're from. And so you got this sort of two unique genetic signatures, potentially unique genetic signatures coming into play. And during this whole time, sorry, I missed the little thing, is that black ducks were decreasing 2 to 3%. So of course, they're like, oh, with the increasing mallards, it's competition for mates and habitat and other resources. And so this became a conservation issue for American black ducks. And so the question was, what is happening in the East? So what is that genetic signature? So to answer that question, I went, it's not in a review, it was published in 2020. Sorry, I haven't updated this. We published this in 2020 and we identified, of course I used Khaki Campbell as my park duck. And then we have these sourced game farm mallards from Kentucky, New Jersey, specific locations where they're being raised and released. And then the wild birds. And thankfully, it was one of those birds, and it is game farm mallards that are being released. Now, why are game farm mallards being released? Well, the dogma has always been that game farm mallards get shot. And if they don't get shot, they are somehow trained not to leave their pond. And if they do leave their pond, they definitely don't survive. If they somehow survive, they definitely don't breed. Well, that is just incorrect, because the only way to have genetics on the landscape is breeding. And so these birds are not only breeding, but they are literally swamping out the wild North American wild signature in the East Coast, and it's starting to creep westward. So of course I had to ask the question, well, maybe this is maybe there's something weird going on, and the East Coast was always just a weird thing. Maybe there was these things. So I went to the Smithsonian and I got historical black duck and historical mallard samples from 1860 to 1915. So before all these mallards were being released, and I did a bunch of genetics on them. So we did some ancient DNA analyses, and this is what we found, is that this is what's called a co-ancestry plot. And essentially it's individual by individual. You're looking at the hotter the color, the more genetics you share with those individuals. And so then we just cluster individuals. And the important thing here is that American black ducks from 150 years ago and American black ducks today, genetically pure American black ducks, are identical. There's no difference. And so if genetic swamping was really the issue here, it didn't happen. And so I think what's happening is that American black ducks, good American black ducks that are around today, majority of them, not mostly the ones in the boreal forest that breed there, are picking correctly. And all those that were picking incorrectly, creating all these hybrids, have dissipated. The other part of this is that historical mallards, again, 1860 to 1915, are identical to mallards found in the Mississippi and Western flyways. And the other part of this is historical mallards and historical black ducks, contemporary mallards and contemporary black ducks have always been closely related. Their history, their genetics has always been very close. Now, here's the kicker, that Eastern mallards today don't group with any of them. They're actually quite the outlier. And so Eastern mallards no longer equal the North American mallard. And so what we've had, what I was able to show in that 2020 paper is that the game farm mallard was released, and it is slowly spreading westward. And I will tell you that the most recent numbers that we have is in the Atlantic Flyway. If you look at a mallard, you've got about a 5% chance that is a wild North American mallard. And one day, I'm sure we'll have a discussion of which one of these European colonists was most successful in North America. Through a variety of different analyses, we were able to recreate. And I have this for Mexican ducks and model ducks, and it looks very similar to this. Is that the North American mallard, the wild mallard, was the ancestor to these monochromatic species. And that all those hybrids that everybody was afraid of are not actually all that there. What is there is that in the 1920s, when people started to put game farm mallards, is that we are transforming the mallard population to now having a wild mallard, mostly in the West, and this feral X-wild mallard hybrid swarm, mostly in the East. But you still have good American black ducks today, and you do still have good wild mallards. And so this, of course, has led my research to now understanding this, from the wild mallard complex to the domestic mallard complex. And if you don't know, the mallard is the ancestor to essentially all of our known domestic lineages, essentially the wolf and the dog. It was domesticated 500 BC somewhere in central China, and there's been a variety of types of ducks that have been created. Of course, everybody knows the Pekin duck. Today I talked about the khaki Campbell, but this is the bird that was developed, created, however you want to call it, somewhere in Europe for, again, hunting and shooting purposes, and has been spread around the world, in fact. And so what has happened is that these birds, these domestic mallards, have been spread. And wherever I study, whether it's North America, Europe, Hawaii, New Zealand, these have interbred with the wild birds that are there. Of course, I'm about whether in North America, in Hawaii, or in New Zealand, while there are still Hawaiian ducks and there's still New Zealand gray ducks, many, many, many of them there's large hybrid swarms in all of them. Today, I'm going to kind of focus on North America, and as this is our largest project to date, and give you a sense of what the scale at which this is causing issues in the gene pool of the wild mallard. So again, mallard history from wild to domestic around the world. So in Europe, Eurasia, this is essentially the origination of all mallard derivatives. Mallards were domesticated about 2,500 years ago, and a variety of types, again, just like the dog, have been created. Game farm mallard strains created in the last 2 centuries via selective breeding for a variety of traits that, you know, shooters enjoy, which would be smaller, faster birds, quicker, of course, quicker growth rates and all these types of things. Today, so they've been releasing these birds in Europe, and unfortunately their populations have declined to the point where about over 3 million game farm mallards are released to sustain their fall flights. In North America, from 1920 to 1916, about 500,000 of these game farm mallards were released annually. From 1960 to today, it's about 250,000, with 210 to 220,000 of these, so the greater majority of them being released in the Atlantic flyway still. The South Pacific, Hawaii game farm mallards were imported in the 1850s for food and commercial farming in the 1930s and 40s, and now there's multiple feral populations currently existing. In New Zealand, similar story, about 30,000 game farm mallards have been imported throughout time, from 1860 to the most recent being in the 1970s. And today, what that has resulted in is a sustainable, self-sustaining feral population of mallards. Of course, I won't be talking about any of those, and so we'll be kind of diving into so all of these birds have been integrated into the world. So what are the genetic consequences from introducing these things? To answer this question, I went out and partnered with lots and lots of folks across the world to get at that. And we have 1,916 samples here. Of course, the majority being from North America across flyways. But of course, a lot, almost 200 out of Eurasia, about 100 known game farm mallards in North America, another 62 from Europe, about 88 feral mallards from New Zealand, another 31 feral mallards from Hawaii. So this is a scale at which we are working with. And again, if you don't know, in North America, because you're going to see these plots, North America is broken up into these 4 flyways: the Atlantic Flyway, the Mississippi Flyway, Central and Pacific. And I'll be showing graphs that in North America identify those flyways specifically. Again, we use DD-RADseq for all of these 2,000 samples, lots and lots of loci, really great sampling. We're going to be working with thousands of loci when we're looking at these things. Before we look at the nuclear part, we're going to look at mitochondrial DNA. So mitochondrial DNA is perpetuated by the female lineage, and it's only provided by the female. So the female gives the same mtDNA to all their offspring. Same thing in ducks, same thing in humans, same thing in all mammals and birds. And so the big takeaway here is that wild mallards, mtDNA haplogroups follow continents. So again, the old world, so your wild ones here in green. In North America, we have it split up into these flyways. And in North America, the majority of them are here in the New World B. There is, however, only one mallard from China that possessed this New World B haplotype. And so that might have been an actual migrant. The domestic and all feral mallards harbor fewer haplotypes and are actually found in these haplotypes right here. This star-like pattern is in fact indicative of what happens during domestication when you go through these bottlenecking events. And in fact, it was this here in black is the Arkaki Campbells, our sort of park duck. And one mallard in Russia that was identified as wild was in fact a khaki Campbell. We found it with the mitochondrial, and you'll see the nuclear assigns it to that as well. So that was nice to find. Over 90% of all the old world mitochondrial DNA in North America can be found in these haplotypes and are explained amongst the major haplotypes that are not only found in North America, they're found in New Zealand, they're found in Hawaii and throughout Europe. And so this really says that all the hybridization and discussion was really a discussion that isn't about wild discussion, but it was actually man-made discussion, which was the fact that we put these birds out. If we put this on a map, just to show you, Old World A haplotypes, in fact, originated in Eurasia. We can see that one bird in China that has the New World B, potential migrant. But then in North America, you have that presence decreasing as you go westward. So the Atlantic Flyway, where we put out the most number of these birds, that is where we find the highest number and rate of these Old World Eurasian origin mitochondrial haplotypes. All right, so that was mom. What about mom and dad? So to look at nuclear, this is a plot, a Pca plot. If you've never looked at a principal component analysis, essentially the dots are moved further away from things that are different and closer together for the things that are the same. And so the clustering tells you something about how similar or dissimilar things are. The first thing I want to point out is that there's a wild cluster, there's a domestic cluster, and then there are these hybrids. First thing, so overlapping clusters of game farm mallards from North America and Eurasia. That really suggests that feral game farm mallards found in Europe and Eastern North America are the same thing, and they in fact are nuclear wise. There's overlapping clustering of wild mallards from North America and Eurasia. So a wild mallard is a wild mallard, except for Greenland mallards that show quite the distinct pattern that we're also looking into further. All remaining domestic lineages have diverged to various reasons. So, you know, mallards in New Zealand, Hawaii, khaki Campbells, they're all under different selective regimes and pressures that would then further cause divergence in their lineage. But no matter what, they are of domestic origins. And wild and domestic hybrids are recovered where the largest number of them are being released. This is those ancestry, before I plot everything, these are those ancestry maps that I've been talking about. Again, game farm mallards, the same genetic stock here and in North America. I know you might be looking at it and it says, oh, the game farm mallards in Europe and the game farm some form of game farm mallards in North America have a bit of wild in them, and that is true. And it is known that European folks like to catch wild ducks and breed them into their lineages. And the ones that you can see in group 2 of game farm mallards, so these guys right here, are from a person from South Carolina that told me that his great granddaddy or granddaddy in the 1980s used to catch wild mallards and breed them into their stock. So indeed he did. Wild mallards, this is the first time that we actually see some population structure between wild mallards and Eurasia in this dark green, and in North America in this light green. Hybrids, these are game farm mallards and wild mallard hybrids are found in the Atlantic Flyway. So you can see lots of Atlantic Flyway mallards being a composition of both green and red. Some in the Mississippi Flyway and fewer as you go west. Feral mallards in Hawaii are actually game farm mallards too. I did not expect that. Number 3 is khaki Campbell, so that black, and then the dark red, which is number 5, is the North and South Islands of New Zealand. So they've actually diverged from the domestic lineage, which was game farm as well. Yeah. So the subsequent analyses that you're going to see are based on these assignments where we say, okay, anybody that has less than 10% interspecific assignment, wild or game farm, is pure parental, and everybody in between are hybrids. So let's plot this out. So this is the plotting, and in fact, we can see the epicenters of where hybridization is highest, and that is in Europe, where Sweden and France are the ones that throw out the 3 million mallards, and we can see that dissipation as we go outward from that location. And in North America, it's east to west. The largest number of hybrids are in the Atlantic Flyway, and that, of course, dissipates as you go westward. Now, in North America, we have almost a state-by-state sampling, so we can actually see some of these cool patterns. And in general, when you look state by state, you see a westward decrease in Old World A haplotype and hybrid ancestry. Again, you can very much see, like, you got lots of red, a bit of red, and that sort of dissipates. in the mitochondrial, but you can really see it in the nuclear where as you get to the prairie pothole birds, you know, you start to see mostly wild North American mallard. I also want to use some analyses to look at, you know, what came first, using some phylogenetic analyses. And essentially we can see that the most basal group, so these wild, is the wild ancestor. In fact, it's the Eurasian wild mallard. And so that is this ancestor to all. Eurasian game farm mallards, it was nice because Khaki Campbell sort of rooted that group, is the ancestor to North America, to Hawaii, and to New Zealand. And the nice thing is that when we tested this, you know, for gene flow and a phylogenetic background, we found significant introgression of wild into that game farm mallard to North America, which is that South Carolina bird. So that's good because we know that that happened. And then essentially wild and domestic ancestors into New Zealand, which is exactly what happened in New Zealand. On top of all this, I wanted to also show you some of the more recent analyses that we're looking for, is that we're trying to understand adaptive differences amongst these groups. And we're using, if you've ever used genetic or niche modeling, this is something we call genetic niche modeling. Essentially, we use a program that uses machine learning to create associations between genetics, genetic allele frequencies, and genetic loci, and the habitat. Various, you know, everything from various indices of environmental indices. And so, and then you can plot that out across across land, in this case, North America, Eurasia, Hawaii, New Zealand, where we have samples. And so what you're looking for is change in color is predicted change in genotype. So you expect a change in color to then correspond, hey, we expect these things to be adapted differently. What I wanted to just point out here is that the environmental factors that we found to be important in important and associated with our genetics is what you sort of expect for ducks. And that would be diurnal range, so the mean diurnal range, temperature, precipitation, and vegetation as being the main drivers. All right, so what do we see here? We see that the same genetic niche space for wild mallards from Western North America and Eastern Eurasia. What's interesting is that feral populations spread out into two genetic niche spaces, so Hawaii and New Zealand, and they're kind of spread out in different locations. And we think that that's actually linked with urbanization. Western Europe and Eastern North America show very similar genetic niche space. And this, of course, is where you have the largest amounts of domestic these game farm mallard introgression. And that is quite interesting. So the adaptive landscape of these birds have been significantly impacted from their wild ancestor. And so populations are showing these local ecological adaptations that appear to be human linked. The other part that we're studying, that we're adding to this is in fact, urbanization indices, because if you ever looked at a light map, Western Europe, so Europe and Eastern North America look like Christmas trees. And so potentially the urbanization of those locations are also impacting the adaptive landscape of these birds. Why is this all important? Well, wild and domestic birds are, hopefully I've convinced you that they're genetically different, but they're also biologically and ecologically completely different. This picture on the top is a common picture I get from hunters, and they're like, oh, that big mallard is, it must be Daffy Duck. And I tell them, no, in fact, the left one, the small mallard, is your game farm mallard, and the right one is actually your wild mallard, the bigger one. The general differences, lots of differences in fertility. Game farm mallards like to basically dump eggs, not watch them. Their growth rates are significantly different. Game farm mallards are 2/3 the size. Build morphology. There's been studies and things that we're doing right now is that their build morphology has been completely changed by domestic setting to eat bread and large seeds that we dump out there, Purina chow. and in fact are completely inefficient at eating wild seeds now. A bit of study in Sweden showed decreased survival. In fact, they only have a survival rate of 4 to 5%. Of course, 4 to 5% of 500,000 or 3 million is still quite a few birds, obviously. Game farm mallard hens in particular lay eggs earlier and for longer, have larger clutches, all the kinds of things that we like in a domestic settings. And of course, here, I hope I should have shown you, game farm mallards are on average 5 to 10% different across the genome. And that is actually at species level if this was actually 2 different species. So, and of course, the adaptive parts that we're really leaning on now, it seems like their adaptive landscape has been reduced. So what does that mean into the future? I guess it all depends on how much urbanization that happens into the future and how they adapt with humans. But of course, what this is also leading to, and what we've already learned in Eurasia, is that stalking is forever. The problem is that when things become domesticated or put into a pen, it doesn't take many generations until many recessive alleles and maladaptive traits that are great in the pen are increased in prevalence, and thus once you put them out, they don't really survive in the wild. And one of my biggest projects right now is to understand exactly what happens when domestic and wild population interact. How does natural selection work on these populations? What potentially we might have to reconsider what the genetic threshold of a pure parental. And to do this, I was thankfully provided NSF funding a year and a half ago. And so we are in the midst of a 5-year study to really understand this, using full genomes to understand of these, how the genome changes as things back crosses, and each generation that they survive, you'd think that natural selection would be parsing out some of these maladaptive traits. We're also working with lots of different museums to go back in time. Thankfully, ducks and duck hunters have been around, and they've been putting them into museums. And so we're going from 1800 to 2020 across space and time to understand them, what's happening there. So with that, I want to acknowledge all my collaborations, obviously sampling efforts across the world, lots and lots of people, all the students in my lab, and obviously all the funding. And for all of you folks listening to me ramble about genetics and ducks. And so I want to thank you all for listening. If you have any questions, you can reach me at that email, look at our website. My students mostly do Twitter. So yeah, enjoy that. And I will stop sharing and take any sort of questions and comments you might have. And I, of course, would be happy to extend my, what we've, we have this kind of data for black ducks and model ducks and New Zealand gray ducks and so forth. So I'd be happy to let you know what we know.
So this is Pete Peterman. I do have a question. As birders, we look at field marks. So it looks to me from your presentation that I can't really tell the difference between a wild mallard, I'm talking the East Coast now, yep, and a game farm raised mallard, or the hybrid. They all appear to be the same unless I put them side by side and say, okay, the wild mallard is larger, has a longer bill.
That's
but
right.
That's pretty difficult in the environment that you're looking as a birder. Am I right?
Yep, that's pretty right. However, we're just finishing up a study to give you those resources. So we just did all of these types of measurements on a bunch of game farm and wild birds to look at exactly what traits we could do. So you're right, many of them are structural. However, I have another student working on an AI application that may be able to, that is working. And for all the mallard-like ducks, because we have so many genetically vetted samples and varieties of plumage types and so forth, that we are hoping that we're going to give you an application that you could either import a picture or take a picture, and it's going to give you sex, age, as well as genetic predisposition. So we are hoping to get those kinds of resources for folks because it's not only just mallards, but like you guys had a discussion. In fact, when I looked at those two black duck pictures, I said to myself, those two actually look pretty good. And they actually look like wild black ducks. Just so happened that there's a mallard there. And it, hopefully, like I have many slides on black ducks and Mexican ducks and what everybody thought were character and model ducks. The characteristics we use are completely incorrect. We thought they were when we did it, but my student, we were basically across all of them, you're 60% correct at actually identifying an individual as a hybrid. And for Mexican ducks, hopefully published here soon, we created a key that's 98.5% accurate for each sex age cohort. They are very different once you figure out what those traits are. And unfortunately, for many of these birds, the traits we picked are actually the formative plumage. It's the first year's bird and we think that maybe they're just not, it's kind of like a teenager. Can't quite control its hormones and it's expressing a bit of ancestry, which is mallard. So it's going to show these mallard traits, and we have them not only characterized for Mexican ducks and model ducks, but also in Hawaiian ducks and in black ducks. In fact, I have pictures of this person that's like, hey, we banded this bird last year. We called it a hybrid. Green in the head, white wing bars. Recaught it, perfectly good black duck. I mean, look, lost the green head, lost everything, it was just, and they had to recall it. And I was like, that story is now across the group. So for you, I would have to be like, if you ask me, like, what is this thing? My first question is, do you think it's a hatch year? Do you think it's a formative bird? In that case, things get a bit tricky. Now, if you've got like, no, that's an adult and it's showing all kinds of crazy stuff, then that's highly likely a hybrid. I mean, we know this if females, right? When they get super old, that old female mallard that starts to show green in the head. That's why we think it's a estrogen testosterone mechanism. That's essentially, they all have the green head genes. It's a matter of suppressing or turning them on. And very young males and very old females have a problem of controlling it, it seems. Hopefully that made sense. It's a complex and complicated story, but genetics has definitely given us finally, when we were able to see the parentals, I was like, all right, time to look for hybrids, and they were not there. I mean, there's few and far between for all these species that people have thought they are being hybridized out of extinction. I will tell you, mallard black, the reason that I think that there's black duck still around is that hybrids tend to backcross into mallard. And so they are not moving mallard genes per se into black duck. Now, whether that's because good black ducks are like, oh, something's wrong with you, and just they don't mate pair with them, and potentially that's also part of a mechanism, but there has to be a mechanism that stopped it. So a bunch of birds incorrectly paired, and now what's left as far as black ducks go, I mean, they're the same exact thing as 1860. So must be choosing correct. Some proportion of them must be choosing correctly.
Yeah, Phil, I was going to ask, because there's a lot of compensatory mechanisms to keep lineages isolated. And it seems, and you show that nice chart with the across the birds, but frequently ducks come up as ones that hybridize frequently. But you have all that, a lot of sexual dimorphism and all those behavioral things. So what's going on with ducks not getting it right? Is it just a matter of I'm super horny and I'm gonna have to settle, or are they just not that particular when?
Yeah, what I taught this morning to my waterfowl course. Yeah, so there's several reasons. So you're right, extra pair copulation quite high in ducks, doesn't lead to a bunch of successful pairings, about 5%. But hey, that's still a lot over a lifetime, right? That's why that trait is still there. The other problem with waterfowl is they often will dump eggs into the wrong species, will leave their brood with the wrong species, and that creates that like bond that's incorrect. So those are definitely mechanisms that result in hybridization. Now, the real question is, so you have all this hybridization, why do you still have distinct species? And something that I didn't show you here, but all of our simulations and this empirical data that we've got that supports it is that as long as you've got a large enough parental gene pool, it doesn't take very many generations for the genes from the other species to basically be swept out. And in fact, it typically takes 3 or 4 generations. And if you think of a generation about a year for most ducks, that's not a lot of time. And so potentially that's why we don't see that swamping effect that we saw in the Atlantic flyway. First of all, again, Atlantic flyway never had a large population, so potentially that's how you swamp them. But that's why it's very difficult to get swamp the prairie pothole bird population that's like, 8 million, right? So that's a hypothesis, and we're testing this. Another example of this is Hawaiian duck, right? So Hawaiian ducks were extirpated from the islands. Of course, feral mallard populations are everywhere. In the 80s, they wanted to reintroduce those populations, Hawaiian ducks in there. Of course, they didn't deal with the mallards. We did a study, and every single one of those populations is a hybrid swarm now. There's not one Hawaiian duck left. And I'm working with Fish and Wildlife to figure out what we can do without starting over, particularly because you can't just kill them all, because everybody likes to feed their ducks and stuff. So it's more a human dimensions issue more than what we can do. But that was a clear case of you use a small population and you've got no more parental to backcross into. Of course, that leads to, what do you do with the Atlantic flyway? I don't know. You don't have much of a parental population left. So it's basically hybrids with hybrids, more or less, at this point, which is kind of crazy. At that landscape level, it's kind of crazy. But that's what we're seeing in Europe as well, because and of course, Europe is having the issues that most fisheries have, is that if they don't stock it with 3 million birds, they don't have very much of a flight. So of course, I have to say now is that where North America wants to head. And then people say, how do you know? And I'm like, I don't know unless I have a time machine, but we could look at what happens to fisheries and what happens to pheasants and bobwhite and everything else we stock. It's the same exact result over and over and over, but that's another discussion.
So I was curious, throughout Europe and some places like Florida, there's a lot of exotic species, especially waterfowl, that are kept. I recognize those numbers are relatively low compared to the native species, but are you aware of how much hybridization or mixed matings might be going on, pairings, because of those anthropogenic factors?
Yeah, it's really the game. So for whatever reason, it's not park ducks that are the problem. There's a bit of that, but that's not, no. Okay, so let me retract that. So in Florida, it is a problem. And that's mostly of Disney World. It's called Hub's principle. If you're around, if that's all you got, that's kind of what happens. And of course, domestic lineages have higher testosterone, have higher propensity to mate no matter what. And so you get a bit of that. And it just so happens that park ducks is what you have in Florida. But the rest of the areas, like Massachusetts, I have a data set of almost 300 across the state, and everything from parks to what they call really good habitat. And it's not park ducks. I don't find park duck in there. It's game farm mallards. So wild birds, might be at a park, but it seems that they don't really hybridize with those types of birds for whatever reason. But with game farm mallards that clearly are their type, they do. And whether it's the forceful nature of game farm mallards or not, I have no idea. But that's who they do. But you're right. I mean, I've been trying to figure out whether other park ducks or these various other lineages are the problem. That's why we have those khaki Campbells. It doesn't seem that that's the case.
So another question, I'm sorry, I got all nerdy.
No, do it.
But so when I was learning things back in the day, one of the things that came up was it was really hard to get DNA from bird skins because they were treated with various things like arsenic and what have you.
Sure.
So how, I know I read your CV, you have background in doing ancient DNA extraction, but how hard I'm curious, how hard is it to actually get viable DNA from Old Museum specimens?
It's actually not that hard anymore. So we go for the toe pad, so that way we don't mess with the skin. Skin stay intact, and we usually like cut out, let's say, this part right here, and then it's been really great. I mean, I've got 2 postdocs at the Smithsonian right now, and they are in charge of about 800 toe pads. So we're extracting DNA. Of course, they're not all going to work, but hopefully a good proportion. And the methods we've got to basically, it's kind of like a fishing expedition now. You can't just like sequence it. You can create these like little baits, and no matter what the fragment size of that gene is, you should be able to catch it like a magnet. And so, yeah, it proved well in that 2020 paper. We sort of like optimized it, and now we're applying it to a whole bunch of mallards across time and space.
Cool. I'm out because I'm an entomologist, right? So I can only read so much ornithology stuff for fun. I just haven't been keeping up with bird genetics that much.
Yeah.
So not to dominate questions, are there any other questions for Phil before we free him to have dinner and enjoy going home?
Yeah, survival rate of the Eurasian mallard, the game farm mallard, I thought you said it was like much worse than the wild mallard in the Atlantic Highway.
Yep. Yeah, I mean, we don't have any here, but the one study out of Sweden found about 5%. And you think like, and yes, you think like that's low, but again, 5% of 3 million or 5% of 500,000 is still quite the large input. And in genetic terms, you only need 1 migrant per generation to basically cause issues.
If we were to stop the game farm production, that would predict, would it not, that they would disappear? And
yeah, so my prediction is exactly the same prediction for salmon stocks and other. You're going to have a crash, right? And so then what's going to be left are things with good genetics and able to survive. And give it a decade or 2 decades, yeah, it'll bounce back. But that's the reason you don't stop stocking salmon. Nobody wants to crash.
you can't stock, you can't raise, quote, wild.
Yeah, so you
genetic
can't. That's a good question that comes up, and yes, you can, but you're never going to get the output, right, that people want. You're not going to make, like, we have a feeding trial going on right now at Forbes Biological Station. And while game farm mallards just basically pooped eggs on the ground and you just pick them up, wild mallards, they get stressed out and they don't want to nest, or they don't want to, you can't make a wild animal do what it doesn't want to do, right? And that was the issue in the 1920s. At least that's what was written, and why anybody would go to Europe, sail a boat to Europe and go get game farm mallards there. Yeah, you definitely can, and I'm sure there's lots of folks that know how to do it with enough space. It's a matter of getting it's legislative, and why they don't even ban these birds, so we know where they're going. It's a political thing, to say the least.
So the hunting organizations that have done so much to create habitat up and down the East Coast, I presume they're out raising mallards too.
Yeah, so it's funny because when I talk to those organizations, again, we were all under the dogma that this isn't a problem of them being out there. But the funny thing is that I tell them, I'm like, you just bought this golf course and you turn it into a beautiful wetland. You actually decrease the carrying capacity of your mallard. The golf course was the correct habitat for that mallard. It's a funny thing because our feeding trials are showing that these birds, both hybrids and just game farm mallards, not only take twice as long to feed, but they need twice as much feed as a wild bird when they're eating wild seeds. If you feed them bread and corn and large seeds, then yeah, all's as well, but their filtering mechanisms no longer are optimized for wild seeds. And so you throw that bird into the wild, you've got a smaller bird, you've got a 700-gram bird that can't feed, and you've got a cyclone that goes through New York. What happens to that bird? You know, it's going to die. Survival goes down quite a bit. And so that like right now it's a hypothesis, potentially at least, you know, partially explaining, these are the terms I have to use, the decline in the mallards. So the US population went from 1 million to 400,000 in about 15 years. And so, and this is a classical boom and bust. I don't know if you know the history of turkeys, but this is essentially what happens. You can put birds out, they boom, and then all of the sort of maladaptive traits increase in enough frequency that you have your bust. And that's the hypothesis right now. So we're working on it.
Anything else? If not, we'll thank Phil, because we had a wonderful evening. I really enjoyed thinking about population genetics and the complex intertwines of, you know, history, human anthropogenic influences, evolutionary glaciation. Like, that's why I became a scientist, and hopefully everyone listening gets excited about the complexities of the natural world out there.
Yeah, I didn't even get to talk about, but basically the glaciers explain all the mallards that are here. And maybe another time I'll be able to sort of explain how that goes. But this is, yeah, it's you know, I was always looking for the hybrids of mallards and all these other ducks, and I found a bunch of mallard hybrids, mallard hybrids. So go figure.
Well, thank you again.
Yeah, thank you. And again, my info's there. Sean, you can pass along the info. Anybody has questions, comments, anything like that, happy to answer.
Certainly. Thank you again. I really appreciate it.
All right. Have a great evening, everybody.
I wish everyone a fair goodbye. Hopefully I see you in the field, and I will catch you on the flip side, perhaps this Saturday. Just for some of the board members, I'm going to send something out so we can think about meeting. And since there's some people here, because we said we would revisit this potential meeting in person starting in March. So just to plant that seed to readdress that, COVID still on the rise and some other things, but just so that little seed is brewing in your mind so we can talk about that in the near future. And if you're a non-board member and you want to weigh in, please feel free to email me, and I'll take those to the board when we have an impromptu meeting in the coming weeks. Again, hope everyone's well. Have a hopefully wonderful Valentine's. If no one loves you, realize I love you, and I'll see you sometime in the future. Bye-bye, everyone.
Better.